Okay, to continue talking about water and the way we were talking about it in the end of the last one, water is very important to the body. It is what allows much of what happens metabolically to happen. It has certain characteristics. Um, Reactivity, high heat capacity. Part of the reason we can actually regulate our body temperature so perfectly, 98.6 approximately, is because of water's ability to absorb and then release heat. Certainly lubrication, water is a high component in the connective tissue that you have with regard to, say, cartilage. In terms of blood is a connective tissue, and water is the high component in water. With that, with the serous membrane, visceral and parietal, the serous fluid is a water based fluid, so this lubrication happens. Mucus has a high water content to it, as well as saliva. Anytime you think when you're lubricating something in the body, there's going to be a high amount of water in that particular lubricant. And then the thing about this polar, non-polar, ions, which would be your sodium chloride, and polar compounds will disassociate in water, so the bonds will be broken in water. Polar molecules tend to be polar molecules tend to be hydrophilic. So they interact with water. We think of them as water loving, whereas nonpolar molecules are hydrophobic. They fear water. They don't react with water. And nonpolar molecules that are in the body are fats and oils, and the cell membrane is a biphospholipid membrane. You'll learn that very soon, and so it has a fat or a lipid aspect to it, which keeps it from disassociating in water, which you don't want that to do with your cell membranes. Concept of pH, and hopefully some of this is review for you. It'll be real helpful for you. pH is the relative concentration of hydrogen ions. So a particular chemical's ability to give hydrogen ions into solution. And if you have equal amount of hydrogen and hydroxy ions, so hydrogen is the H plus, hydroxy is the OH with the negative on it, that would be a pH balance, and water tends to be our standard with regard to neutral. Water is a pH of 7. Acid is anything lower than 7, so if it's 6.5 on down to the end of the scale in the pH scale, it's acidic. Basic is higher than 7, and human blood tends to be pretty neutral, 7.35 to 7.45, and that is a one of the more constant things that that go on in the body is the pH of blood is held at that range, and many things happen in terms of the buffer systems that go on in the body to keep that pH. Just a kind of an FYI, it's an inverse relationship of hydrogen concentrations, which means the more hydrogen concentrations, the lower the pH, or the more acidic something is. Fewer hydrogen concentrations, higher the pH, more basic something is. And here's your image, which is really wonderful, because once you know what you're looking at, it really makes a whole lot of sense. We think of acid as red. There isn't anything red about acid. It's just the color we've ascribed to it, and basic as blue. Again, just arbitrary colors we put together with it. And so you have neutral, you have 7 in the middle, and then as you go closer to 0, you're becoming more and more acidic. As you go closer to 14, you're becoming more and more basic. A few things in there. You have stomach acid is very acidic. Saliva is a little acidic, and household bleach, ammonia, very basic. Oven cleaner, extremely basic. And so they have some chemicals and where they lie on the pH scale. Organic molecules, as we said before, have hydrogen, carbon, oxygen in them usually. They're covalently bonded, meaning they share the bonds, and they consist in the body anyway. Important organic molecules that we are interested in as humans are carbohydrates, lipids, proteins, and nucleic acids. Proteins are amino acids and nucleic acids. We'll start with carbohydrates. Carbohydrates can be single bonded, so they can be monosaccharides, they can be double bonded. So you have a simple sugar that is bonded with another simple sugar via this dehydration synthesis reaction. Remember, that one is the one that you're going to take water out and make a disaccharide. Polysaccharide are many monosaccharide units condensed into a long chain. And then you have glycogen, which is considered starch, and cellulose. So these are types of carbohydrates that are utilized by the body. Monosaccharides are absorbed or utilized quickest, because if you start out with a polysaccharide, it has to be broken apart and then turn into at least a disaccharide. But a polysaccharide can be long strings of monosaccharide units. So whatever you start out with, it has to be broken down into a monosaccharide in order to be utilized by the body, utilized by the cells for energy. And this is the image from your book. Again, sometimes it's good to hear it, read it, and then see it. And in the seeing it, then you get an idea of, okay, that's what that looks like. The image B is your classic structural formula of glucose, and most of you have probably heard of glucose. Glucose is what we think of as our basic energy unit in terms of a carbon dioxide, and there you have it chemically written. I really won't ever ask you anything about the chemical writing of these organic or inorganic molecules. However, sometimes having that in your head will help you understand, okay, that's how the body uses that. And then you have this whole thing about the dehydration synthesis, just to kind of review that reaction. So you have glucose and fructose, and those are all monosaccharides. And we are going to take water out, so we're going to hydrolyze, we're going to take water out and bond those together. So we are going to synthesize. I just said that wrong, I guess. Hydrolyze is going to put water in. So what we're doing is we are taking two individual monosaccharides and building up. So this is an anabolic reaction. We're building up by creating a water molecule at the end. So dehydration synthesis. Dehydration, that kind of makes sense. You're taking water out. And then you have your hydrolytic reaction, where you have this sucrose, which is the disaccharide, putting water in the mix, and then breaking it apart into two monosaccharides. Lipids are very important in the body because they're hydrophobic. They are afraid of water, I guess you can say in very simplistic terms. They don't disassociate in water. They don't react. They're non-polar. They don't react with water. They just exist with water. And in your head, you can think of, if you pour oil in water, it just turns into little bubbles. It doesn't mix in. You can shake it up as much as you like, and it turns into little droplets of oil, but it never changes its nature. It can become smaller and smaller droplets, but it's still going to be the same thing in water. And this very important with regard to anything that's in the body that we don't want to disassociate. We don't want it to break apart. The oils, lipids are fatty acids, eicosanoids, sorry, glycerides, steroids, phospholipids, and glycolipids. What phospholipids and glycolipids are you're putting a phosphate group with lipid group, glycolipids, glycogen with a lipid group, and you have different properties because you're putting those together. You'll hear more about those when we talk about the cell. In terms of fatty acids, you have long chains of carbons and hydrogens, and at the end, you have this carboxyl group. Mostly non-polar, except for the group hanging off. That makes them kind of unique. They're mostly non-polar except for this group hanging off. And what you can have with fatty acids, you can have saturated or unsaturated. So saturated hydrogen, no covalent bonds, or unsaturated where you have some double bonds that actually can be broken pretty easy by the body. So that you've probably heard about saturated and non-saturated fats. The reason non-saturated or unsaturated fats are better for you, they're digested easier. Saturated fats not broken down because they're much more stable. And then I should really learn how to say this word, and this is a good way for me to say I apologize. And what you do is you practice them. Eicosanoids, and they're derived. The important thing about these are leukotrienes, and actually you hear it more in the terms of leukotrienes anyway, where you have many small chemicals that are made by the immune system that work to combat or to mediate reactions, particularly when we think of the inflammatory reaction which is going on throughout the whole body. Prostaglandins are local hormones, and what they mean by local is maybe between cells, so communication between cells, or communication in a very small area in the body, so maybe a couple or 3 cells apart, prostaglandins. And you find there are many prostaglandins. They tend to call them prostaglandins A, B, C, D, etc., and they're still discovering even more prostaglandins. It's a form of communication in small regions. Glycerides are fatty acids that have a glycerol molecule attached. Triglycerides are stored form of fat. With triglycerides, you have 3 fatty acids and a glycerol. And this is our energy source, insulation, protection. This is otherwise known as our adipose tissue. The adipose tissue, you have adipose cells and you have triglycerides stored inside those cells. Very important for insulation, maintaining body temperature, energy source or stored form of energy, and also fat protects organs. You have a nice heavy-duty fat pad around the kidney. You have fat around the spleen. It's a good protector of organs. Phospholipids and glycolipids, they will either change the polarity, particularly if you have a biphospholipid layer in the cell membrane. So you have a hydrophilic side and a hydrophobic side. Or the other thing is that glycolipids create kind of a sticky or a unique, that glycogen, a sticky portion of that particular molecule. So when something floats by in the water, it actually attaches to it. Both of these are associated with the cell membrane.